Hello and welcome to today's webinar featuring Rebecca Saxe. I'm Claire Cameron, Engagement Editor at Spectrum, the home for autism research, news and analysis. We'll begin the presentation in a moment, but first, know that we'll be fielding questions at the end of today's presentation and that you can ask them at any point during the session. To ask a question, submit them via the chat window to the left of your screen. And as always, I'll note that if there are any members of the press tuning in, you can only report information presented during this webinar if that material has already been published elsewhere or if you have first obtained express written permission from the presenter. Today's presenter, Rebecca Saxe, joins us today from Cambridge, Massachusetts, where she's currently a professor of cognitive neuroscience in the Department of Brain and Cognitive Sciences at the Massachusetts Institute of Technology. She is also an associate member of the McGovern Institute for Brain Research. Saxe received her bachelor's degree from Oxford University and her PhD from MIT. Prior to joining the faculty at MIT, Saxe was a junior fellow in Harvard Society of Fellows. Saxe has authored more than 60 peer-reviewed papers on the cognitive neuroscience of social cognition, theory of mind, and moral judgment. Her TED Talk on how we think about other people's thoughts has been viewed over 2.5 million times and translated into 33 languages. Dr. Saxe's work uses various approaches, including functional magnetic resonance imaging and other brain scanning tools, to investigate the brain regions that underlie theory of mind and the mechanisms by which the brain constructs abstract thoughts. Welcome, Dr. Saxe.
Thank you very much, Claire. This is a new experience for me, and I'm looking forward to trying it. I've never given a webinar before. So today I thought that I would talk about neuroimaging of theory of mind. The basic idea here is to ask what we can learn about how human brains engage in high-level control processing, how we think about one another's minds, and whether knowing about that neural mechanism in healthy adults will give insight into the origin of social deficits in autism, and ideally in the long run, insight into prospects for treatment. That's the big vision and the long-term goal. What we're going to accomplish in today's talk is more modest than that. So I thought I'd just start with sort of an overview, and the overall structure is a little bit historically minded. So the first half of the talk will be about how researchers, my lab and others, approached the question of studying theory of mind in the human brain from about 2000, when this topic became possible to study, until 2010 or so. And again, the main content here will be an overview of what that taught us, showing us human cortical regions involved in theory of mind, and then I'll talk a little bit about how that could be and has been applied to autism, including dissatisfaction with where that has got us. And so then I'll go to a new approach, new technique in neuroimaging that is increasingly taking over most analysis of neuroimaging data in humans, called multivariate analyses. I'll focus on explaining the kind of core underlying ideas of multivariate analyses, how they work and what they're getting at. I'll show some of what the potential is of these tools to teach us more about neural mechanisms of theory of mind in typical adults, and then a hint that this could be promising as an application to autism. So that's the plan for today. I'm also typically always open to trying to take at least comprehension questions, if not any questions, during the talk, so I'll try to keep my eye on the chat window to see if anybody is confused in ways that I can help resolve. Okay, so that's the plan. And the first place to start is, what do we mean by theory of mind? So I said I'm going to talk about how we can use functional neuroimaging methods to study the neural basis of theory of mind. And that, of course, starts with, what do I mean by theory of mind? So what I'll do is show you a kind of key example of what the task that I'm going to use, and talk about how that is theory of mind and how it relates to other ideas about what theory of mind is as a technical term. Okay, so the kind of prototype example that I give of theory of mind as an example of high-level human social inference is a story about Grace in a chemical factory. So Grace is on a tour of a chemical factory, and she's going to make a cup of coffee for another girl. Next to the coffee machine, she sees a jar of white powder labeled sugar, so she thinks that the powder is sugar and puts some of that powder in the coffee. Actually, that powder is a dangerous toxic poison, and when the other girl drinks the coffee, she dies. So then we can ask, how much blame does Grace deserve for putting the white powder in the coffee? And compare judgments in that case to judgments in a kind of closely matched control case. So again, Grace is on a tour of a chemical factory. She's going to make a cup of coffee. Next to the coffee machine is a jar of white powder. The jar is labeled dangerous toxic poison, so Grace believes that the powder is poison, and she puts some in the coffee. When the other girl drinks the coffee, she dies. Now the key thing about this comparison is that pretty much everything that happened in the world is the same in these two cases. So in both cases, Grace made a cup of coffee, she put white powder in it, the powder was poison, and the girl who drank the coffee died. And so if your intuition is that nevertheless how much blame she deserves, the moral judgment of this action is very different in these two cases, that shows that you're paying attention to and considering as relevant not just what she did and how much harm she caused, but what she thought she was doing and what she should have known she was doing. And so that is a standard task that we've used now for a long time as a measure of how much people pay attention to and consider as relevant other people's beliefs and intentions and desires when making inferences or judgments about their actions. As I said, in this case, one of the standard questions we might ask is how much blame Grace deserves for putting the powder in the coffee. And when we ask this question, for all possible combinations of what Grace thought she was doing and what harm she actually caused, and what you can see here is that both of the cases where Grace believed that what she was putting in the coffee was poison are considered very blameworthy, whether it was actually poison and the girl died, or whether it was a mistake, it was actually sugar and nothing bad happened to the girl. And similarly, both of the cases where Grace believes that it was sugar are judged to be not very blameworthy, even though in the accidental case it was actually poison and the girl died. So we can use this big difference between moral judgments that are based on beliefs as one metric of people's use of mental state information in making social inferences. Okay, so that's an example of what I mean by theory of mind. And that example suggests sort of a prototype concept, but obviously people's use of the term theory of mind extends to many other kinds of tasks. So for example, some researchers have been also interested in the perception of simple motions as biological or goal directives. This is a frame from the famous Hyder and Simmel movie, and have viewed the human capacity to perceptually detect agents and goal-directed actions as part of the same cognitive capacity as the ability to use mental states as relevant in judgments like moral judgments. At the opposite end of the extreme, some people have tried to study comprehension of really difficult mental state attributions. This is the apocryphal sentence attributed to Alan Greenspan, which is, "I know you think you understand what you thought I said, but I'm not sure you realize that what you heard was not what I meant." So these kind of very convoluted, multiply embedded mental state attributions. Both of those, to me, are on the periphery of what we might consider the core competence of theory of mind, and I'm more interested in core or central cases, the basic ability that human adults have to recognize what other people think, want, and believe, and to use that information to predict, explain, evaluate, and coordinate with other people's actions. Okay, so that's the core competence. And now the question is, can we study how that works in the healthy human brain, and if so, how? So this, as a question, arose in the early 80s on the basis of disproportionate deficits in autism. Researchers noticed that children diagnosed with autism showed disproportionate difficulty in basic theory of mind tasks compared to their ability to do similarly complex logical tasks. And that led to the hypothesis that maybe there was some distinctive neural mechanism necessary for theory of mind tasks that could be disproportionately targeted by whatever the etiology of autism turned out to be. That hypothesis remained hard to test until non-invasive neuroimaging was possible relatively routinely. And so in the late 90s and early 2000s, once fMRI was easily accessible to many researchers, the idea sort of arose independently in many labs simultaneously that fMRI would be an ideal technique to ask whether any brain regions are selectively or more required for solving theory of mind problems, for making inferences about other people's minds, than for other kinds of similarly logically complex cognitive tasks. So I, and again, many groups tried this simultaneously, had the idea to try to get people who were lying in an MRI tube to be thinking about other people's thoughts and beliefs. And the way we chose to do this was through having them read short verbal narratives. The first experiments that we did around 2000, 2001 had participants lying in an MRI machine and reading short stories that described actions that hinge on a character's beliefs, so that you couldn't follow what would happen in the story unless you were paying attention to what the character knew and believed. And so here's an example. We could say Anne made lasagna in the blue dish. After Anne left, Ian came home, threw out the lasagna, and made spaghetti in the blue dish, and then placed it back in the fridge. At that point, we can ask what Anne thinks the blue dish contains. And to get the right answer there, you have to consider not only what the blue dish actually contains, which is spaghetti, but also what it last contained when Anne saw it, which was lasagna. So we had participants in the scanner reading these stories and thinking about Anne and her thoughts. And then in the traditional approach to fMRI at the time, what we wanted to ask is, when people are thinking about Anne and her belief about the dish, we know that many, many, many regions of their brain must be engaged. In fact, practically the whole brain, right? You need visual cortex to see the visual stimuli, the words. You need all of the parts of the brain you need to read, to recognize those words, to make sense of the syntax and semantics of the sentences. You need executive function and working memory to hold the story in mind, to choose between two possible answers to the question, spaghetti versus lasagna. You need motor control to push a button, and so forth. And the hypothesis that we were interested in was, over and above all of those capacities that you need to do this task, are there any brain regions additionally recruited particularly because this task requires you to think about a character's thoughts? And the way that we set out to ask that was by designing a control condition, so stories that people would also read, that were matched in terms of all of their other demands, but didn't require thinking about thoughts. That's the goal. The idea is a minimal pairs design. And one of the conditions that we tried at the time, inspired by work in developmental psychology, is called a false photograph control. So here's an example. You read about a Caribbean island where a volcano erupted 3 months ago. Now all there is lava rock, but there are photographs showing the island before the eruption. And we can ask how the island looks in the photograph. Again, to solve this task, you need to see the stimulus, you need to read and understand the sentences, you need working memory and executive function to choose between two options, you need motor control to push a button, but now there are no people in the story and no beliefs. And so we used this design to ask whether any brain regions, as measured by fMRI, show systematically increased blood oxygen when people are reading stories that include a character and her false beliefs, relative to stories that are matched in length and complexity and difficulty, but don't have people or beliefs in them. So this is our implementation of a traditional false belief task for people lying in an fMRI machine. And the idea here then is once people are lying in the machine reading these stories, simultaneously in each position in their brain, we can track blood oxygen over time and look for places in the brain where the blood oxygen increases more when the story has people and their beliefs in it compared to control stories that don't have people and their beliefs. Again, this is a very traditional approach to fMRI data. So then we can look averaging across all of the stories that have beliefs in them compared to all of the stories that don't, and look for brain regions where blood oxygenation is higher if the story contains a belief. So as I said, we and many other labs did this around 2000 to 2003. And amazingly, every lab in the world that tried this experiment at the time and since got the same answer, which is not a given in fMRI research, which is that there are a group of cortical regions showing robustly greater blood oxygen signal when stories describe a character's beliefs compared to these control stories. One of those cortical regions, the one that I'm pointing to in this slide here, is a region that was named the right temporoparietal junction because it seemed to be between the temporal lobe and the parietal lobe in the right hemisphere, although that name is unfortunately very vague. And in this region in particular, if you look at the percent signal change over time, so now I'm going to show you the average, this is just the selective average response, so the time course in this brain region averaged across time, where 0 on the x-axis here is the time when the story is initially presented on the screen, and 10 seconds later is when the question is presented. Percent signal change relative to a resting fixation is on the y-axis. And so when the stories describe beliefs, what we saw is that the blood oxygenation starts to rise almost immediately and reaches a peak while participants are reading the story and stays high until the participants read the question and then falls back to rest. Whereas if the story describes a photograph or a map, so has no person in it, the average response in this brain region is 0, as if we had put nothing on the screen at all. There's a number of things to say about this response pattern in time. One thing is that it tells you that this response is to initial comprehension of the story, so thinking about the story as you're reading it, not to the demand to respond to a question. And we've confirmed that over the years in many ways, showing that it almost doesn't matter what question you ask at the end of a story, as long as the story engaged consideration of a character's mental state. Another thing that's evident from this plot is that this brain region is not a so-called default mode brain region, in the sense that a default mode brain region is a brain region that does not respond, so that responds greater at rest than at externally oriented tasks, and deactivates relative to rest during externally oriented tasks. And here's a brain region with a classic, robust, positive hemodynamic response to its preferred stimulus, in this case a story about beliefs, relative to approximately 0 hemodynamic response to the control condition. So that's just 2 of the things you can tell already by looking at this plot. Now, as I said, it's not just one cortical region that shows this profile. There's at least 5, and many of them are shown here. So right temporoparietal junction, there's also a region in left temporoparietal junction, a number of regions, 2 or 3 different regions in medial prefrontal cortex, and again, at least 2 regions in posterior cingulate and medial parietal cortex. These brain regions respond very similarly to this task, although somewhat differently across more subtle manipulations. And some of that will come up in today's presentation, but I'm also happy to answer questions about the differences between these brain regions at any point. Okay, so that is the the basic profile. Another thing to notice about this profile of activation is that it is just incredibly strong a signal. The 1% signal change, that's similar to what you would get in a primary visual or motor area to a basic stimulus. This is a very robust response. And correspondingly, we can see this response in each individual subject that we scan. So this is just 3 individual subjects to show as an example that participated in this task. And you can see 2 things looking at these functional responses in their native space. One is that activation in the right TPJ, which is what I'm showing here, is a very reliable feature in each individual participant, and that is true systematically for hundreds of participants we've now studied. And the other thing is that there is individual variability, for example, in both the size and position of that activation, and that is also true across a much wider set of studies. But this fact that this activation pattern is so robust in each individual person that you can see where the right TPJ, for example, and all of these cortical regions, you can see where they are after just a few stories about beliefs and a few stories about photographs by subtracting the bold responses, led us to adopt a technique which is I'm going to apply in all the research I show you from now on, which is called a functional region of interest analysis, where for each subject we first have them read stories about beliefs and stories about photographs, and we find in each person where their brain region is that responds more to beliefs than photographs in each of these cortical areas, and then we define that as a region of interest for any subsequent study. So for people familiar with visual neuroscience, I think of this as like the retinotopic mapping of the social brain. This tells us where we are, and we can use that from now on. Okay, this approach as a mechanism for defining individual functional regions of interest is easy and robust and takes only 10 minutes. So if you are doing fMRI research, this is a very easy technique to apply. It works in more than 90% of individual subjects, but after 10 minutes you can define each of these individual regions. And we make the stimuli and presentation mechanism freely available on our website. So feel free to use it if you are interested. Okay, so now we have a brain region that is recruited robustly, or a set of brain regions, around 5, recruited robustly when people are thinking about beliefs and not when people are thinking about photographs. And the obvious question is, okay, that's just one property of these brain regions. Can we get a better sense of what these brain regions really do and what they are responding to in our stories? So the stories that I showed you are very complicated. They have a lot going on. There's many differences between the stories about beliefs and the stories about photographs. And so the traditional fMRI question, and the one I'm going to start with, is a question about something like selectivity. So we could say, okay, across a whole range of different tasks, when are these brain regions engaged? And how can we define the set of things that do versus do not engage these brain regions? That's the kind of question of traditional fMRI analysis. And the way that we and many other people approach that answer is over about 10 years, so from around 2000 to around 2010, we tried having people read many different kinds of stories about all kinds of different information, and also not just stories, also watch movies and see photographs, and we tried to carve out the category. What is it that these brain regions do? What do they respond to a lot, and what do they respond to not that much? That's the traditional fMRI question of selectivity. And I'm going to show you just 2 examples from my lab of how we approach this. Again, this is 2 examples from dozens or hundreds of such studies. So in one of these examples, what we asked was, can you say that these brain regions are really responding to thinking about a person's thoughts? Or are they responding to reading a story about a person in a much more general way? And to test that, we wrote stories. So each unit is a story, but inside each story are 3 parts. One which describes the environment of the story, one which describes a character's internal but physical sensations like pain or exhaustion or hunger or thirst, and a third section which describes the character's thoughts, beliefs, plans, and intentions. And the question was, could we see activity in any of these brain regions that was temporally specific to just the part of the story that was focusing attention on the character's mind? And so to give you a sense of these stories, here's one example. This is a story about Matthew. He's scrubbing away stubborn slime until his arms are numb and his fingers feel spongy. So that's a description of how he feels on the inside, but in a physical sense, how his body feels. We could also tell you about his mind. He thought he would get a cleaner as soon as he could afford one, and about his environment. The kitchen sink is piled high with dirty dishes. So we had 72 stories like this, each with these 3 different parts, and critically the parts could be presented in any order so that we could look for whether brain activation was for anything about a person, anything about a person's internal states, or somehow more specific just to a person's mind. So for stories in this order, for example, when we looked at the time starting when we presented physical information, there was no response. When we added mental information, you get a robust response. And then when we put information about the environment, much less response. And as I said, these stories are designed so you can present them in other orders. So here's a different story in a different order. So focusing on how Pamela feels carrying her 40-pound backpack, the environment, what's in her backpack, and then her mental state. She noticed she hadn't zipped her backpack. And again, you can see that the activation, this is from the right TPJ, is specific within the story to the time when we're presenting information about a character's mental state. Okay, so examples like these over many years started to make us believe that the magnitude of activation in this brain region was predicted largely by whether or not the participant was thinking about a character's mind as opposed to thinking about other things like a character's body or the physical environment of the story or another representation. Okay, so that's one approach we took and other people took as well. Another approach you can do instead of looking within a single story is write many stories that vary on many dimensions and use that to try to isolate a relevant dimension. So in another experiment, we had participants in an MRI machine read 144 different short stories that vary in many ways. So for example, here's a story about Larry. This is a pretty neutral story focusing on Larry's feeling while he's drinking juice. Another story is about Oscar doing dishes and cutting his hand on a sharp knife that burns in the dirty soapy water. A third story, Maggie's in love with a man and wishes he would reciprocate her feelings. One day this man sends a text to Maggie he meant to send to someone else, saying he does not want to see her again. Maggie gets the text and starts crying. So that's focusing on her mental states. So for each one of these 144 stories, we had independent participants on Amazon Turk judge many different properties of these stories. And then we asked how the Amazon Turk judgments of properties of each item predicted the pattern of activation that that item would elicit in the brains of separate participants in the scanner. So for example, one question we asked on Amazon Turk was how much physical pain was the main character experiencing? So this story gets, for example, very high ratings for physical pain on the story about Oscar, pretty low on the stories about Larry and Maggie. And that allows us to ask, okay, when a story focuses your attention on a character's body and physical pain, what pattern of activation does that recruit in the brain? And in particular, these brain regions that we think might be engaged for theory of mind, are they also engaged for these very salient, attention-grabbing stories about other people, describing other people's physical pain? So first we can ask that second question. We can look at how much the story described a character in physical pain and whether that predicts the response in the right TPJ of other subjects. And the answer is that it doesn't. The stories that, like the story about Oscar, that describe high levels of physical pain don't, on average, get high responses in the right TPJ. And instead, if you look at where they do get high levels of responses, it's in regions like the middle cingulate cortex, the anterior insula, and secondary sensory cortex, so all brain regions that also show strong responses if the participant themselves is experiencing pain. So this shows that this analysis is working. It can show us robust recruitment in pain-related brain regions when stories describe a character's physical pain, but not in these brain regions associated with theory of mind. Okay. Another question we asked is about how vivid the scenarios are as a proxy for how attention-grabbing they are. And again, that doesn't predict activity in theory of mind brain regions. The question that does predict activity in theory of mind brain regions is how much did reading this story make you consider the thoughts of the characters? So when we look at each individual story, so each dot here is one of the 144 stories, what you can see is that stories that were rated as making readers consider the thoughts of the characters by readers on Turk were the same stories that evoked robust activation in the right TPJ in participants, different participants in the scanner. And in fact, this is true of all the brain regions in the theory of mind network that I showed you. And one thing that I think is cool about these data is that it shows us that our brain treats stories about other people's minds and stories about other people's bodies really differently. So we get totally different patterns of activation in the red when a story makes you think about a character's thoughts, and in the blue when a story makes you think about a character's body and especially their bodily pain, which is fascinating because obviously other people's bodies and other people's minds are deeply connected, and yet our brain seems to parcel the world into two dualist camps. Okay, but so in brief summary of a decade of research, really a decade's worth of experiments asking the question, what gets these brain regions going? When do they respond a lot, and when do they respond a little? Kind of focused on stories that describe or make you think about a character's thoughts or beliefs or desires or plans, compared to stories about the physical world and stories about a character's other features, physical description or bodily states. And that was the basis for this idea that, okay, well maybe these brain regions' bold signal is reflecting theory of mind, consideration of another person's mental states. Now one thing you always worry about when all of your data come from fMRI is that that is some kind of epiphenomenon or correlation, because all fMRI data are correlational. And so one important part of this story was a check on whether these brain regions are also causally necessary for considering other people's mental states. And we have done one such check. The way we did that is we had people making moral judgments of actions done based on false beliefs or true beliefs, like the grace scenario that I told you about at the beginning. And we had people making these judgments either in a control condition or immediately after transcranial magnetic stimulation to the functionally defined right TPJ in their own brain. And we found that transcranial magnetic stimulation to this brain region changed people's moral judgments in the predicted direction. That is, they made people's moral judgments less sensitive to what Grace thought she was doing, and therefore more sensitive to what actually happened, whether the girl actually died or not. And this change is specific to TMS to the right TPJ. So putting together everything that I have said so far, what you get as an overall picture, and again this is where we were around 2010, is the hypothesis that these brain regions in general and the right TPJ in particular is a brain region involved in theory of mind in healthy human adults. That something about this brain region makes it be active whenever people are considering other people's minds and thoughts, and that TMS to this brain region interferes with our ability to consider other people's minds and thoughts and use that information as relevant for moral judgment. Okay, so that was part one. How should that be applied to autism? Well, so as I said, the original reason that this research started in the very beginning was the hypothesis that autism reflected a disproportionate deficit of high-level social cognition, and that therefore maybe the etiology of autism was some insult to a neural mechanism for theory of mind or high-level social information processing. And so at this stage, as we were getting these results, it seemed like neuroimaging ought to be an extremely useful tool for understanding the etiology of social deficits in autism. And this is really important, right? Disproportionate deficits in social interaction and social communication relative to, for example, IQ, are the basis of the differential diagnosis for autism as opposed to other neurodevelopmental disorders. Because this is the basis of the differential diagnosis, deficits or difficulties with social interaction and social communication occur across the spectrum of autism, and are often the most consequential both for caregivers' experience of giving care to these individuals and for the individual's own quality of life. So understanding the mechanism of social deficits in autism and being able to treat them would be really important. Cognitive neuroscience seems like it should be a key level of analysis because it ought to be sensitive not just to whether people get the right answer, but how they process the social world. And so starting, again, about 10 years ago, the hope was that cognitive neuroscience, or fMRI, would reveal what the basis was of social deficits in autism. For example, the hypothesis that I had around 2004 that maybe individuals with autism process social information using compensatory mechanisms rather than using the typical brain regions. Okay, so that hypothesis predicts that we should see activity in different brain regions when people with autism engage in theory of mind tasks. And we and many of other people have tested versions of this hypothesis, and to summarize an enormous literature, my summary is that although there have been many, many, many attempts to test this hypothesis, these kinds of studies find either no group differences between groups with autism and healthy controls, or small, variable, and basically not replicable differences across labs and groups. Just to give one example, the false belief task that I showed you at the beginning, over the years we have run this exact task on many, many people. And so as of a few years ago, we had run this basic task on over 30 participants with autism and over 450 typically developing adult participants, which gave us the power to really test what brain regions were recruited and to what degree in this task. And both when we look at the full distribution of response, so this is the typical distribution of the size of the right TPJ in typical participants, and then with the red dots are the participants with autism. Both in the full sample where we can look at the whole distribution and in a subset of the population matched for many different things, including age, IQ, etc. The overall picture of this experiment was that there are basically no differences, no measurable differences in the magnitude, size, position, reliability, homogeneity of the activation in this example of a theory of mind task in a group of participants diagnosed with autism compared to in a group of typically developing adults. Okay, so that is pretty upsetting because that is evidence against the hypothesis, or either that there is that cognitive neuroscience, well, it shows that cognitive neuroscience is not sensitive to whatever is the difficulty that people with autism are having in processing the social world, or at least that this kind of approach was not sensitive. So what to do? And now the state of this field is that people are taking multiple different approaches which are complementary and all of which may help and hopefully will help to give us insight into what is the actual neural mechanism underlying deficits in social interactions. Three core themes of these approaches are: one, define more homogeneous and smaller subgroups of individuals with autism. The second one is trying to develop more naturalistic tasks and stimuli on the theory that maybe reading stories doesn't evoke all of the elements of social cognition that are impaired in autism. And the third approach, which may be less well known, and so it's the one I want to talk about today, is using other techniques to measure neural processing in social tasks in autism. Okay, and so the technique I want to talk about is multivariate analyses. So here's what a multivariate analysis as opposed to a univariate analysis. So in traditional fMRI, this is what I've been talking about so far. You have participants read a story. This is about Albert advising his girlfriend to walk over thin ice. Then another story, here Grace is deliberately poisoning a girl who's annoying her. And as the participants are reading, you're measuring the magnitude of response on average in a whole big chunk of cortical tissue to those stories as they go on in time. And as I've illustrated here, both of these stories would in principle evoke large responses in any theory of mind-brain region, since they both evoke consideration of the character's mental states. But in a sense, that is not what we want to be able to study. We don't want to be able to study just that both of these stories evoke thinking about other people's minds, but the actual inference that people are capable of making of who thinks what and why they think that and what the consequences are of those thoughts. Like, for example, that while Albert is being an nincompoop, Grace is a murderer. Okay, so how could we use neuroimaging to get at these subtler dimensions of inference? That is what we hope multivariate analyses will allow. So the basic idea in a multivariate analysis is that instead of looking at the shared large average response of an entire cortical region, you look instead at the much smaller, subtler variation in the spatial pattern of response. So over and above this large shared response, which voxels show relatively more and relatively less activation on a trial-by-trial or condition-by-condition basis. And the key idea is that even though there's a large shared component of response, that these relative patterns of activity across voxels might make an analysis sensitive to differences in the neural populations that are being driven by each of these different stories or story conditions, and might therefore get us closer to the neural processing that supports real inference in these tasks. Okay, so that's the idea. How would it work? So there's lots of reasons to think this could not or should not work, right? It's asking for spatial sensitivity in a tool that does not have that much spatial sensitivity. And so to test whether or not this could work at all, whether fMRI patterns of activation could make us sensitive to subtle differences, we went back to our trusty test case, accidental harm. And the reason we chose this as a test case is that, as I said, we have reason to believe that information about whether harm was caused accidentally or not is encoded in the right TPJ, because TMS to the right TPJ changes moral judgments of these scenarios. But we also knew that the average amount of activity across that entire region is similar for accidental and intentional harm, since both evoke thinking about what the character thought they were doing. And another thing that's nice about this paradigm is that the scenarios are different in a very tiny way. So even though these are complicated stimuli, the condition difference is created with a tiny manipulation. So on a given trial, you would read a story about a character. Here's a character cooking dinner, considering and adding peanuts to the dish, even though one of the dinner guests is severely allergic to peanuts. And then at the very end of the story, we add the critical information, which is whether the character knew or didn't know about the peanut allergy when they added the peanuts. So here we've just changed 2 or 3 words in each individual story, and that changes the moral judgment quite dramatically. So the question is, can we then look at the pattern of activity evoked by these stories and learn something about the inference that accidental harm is different from intentional harm. Okay, so how this analysis goes, we use the localizer task to find series mind brain regions in each individual participant. We measure the response to different stories across a range of actions that are done intentionally and across a range of actions done unintentionally. As I said, we know that the average response in this region is high to both kinds of stories, but what we're looking for is whether the pattern or the relative response per voxel is systematically different for intentional versus accidental harms. And the way we test that is by asking whether, if in a new story that you know nothing about, all I show you is the pattern of response, can you guess from that pattern whether the action was done accidentally or intentionally? So you don't know what action it was, you don't know who did what to whom, but maybe you can tell just from looking at that whatever it was an action that caused harm knowingly. Okay, so if you got that intuition, then you're doing multivoxel pattern analysis in your head. And that is the basic analysis strategy that we used to ask whether the right TPJ contains information about the difference between intentional and accidental harm. So I will go quickly over this since apparently I'm running out of time. So we did this in a first experiment where we found significant but a very small difference in the pattern for intentional versus accidental harm. We then replicated this in 2 additional experiments. So now what I'm showing you is data from 3 different experiments with 3 different manipulations of accidental versus intentional harm. And each time, now across 3 separate groups of participants, we could use the pattern of activation in the right TPJ to tell whether a story was about an accidental act of harm or an intentional act of harm. This information was only present in the right TPJ, so looking across other brain regions associated with serious mind, they did not contain any information about the difference between accidental and intentional harm. But the thing perhaps that I was most excited about these data was if you then look in individual participants at their moral judgments of these stories. So for each person, how much worse did they say the intentional harm was compared to the accidental harm? So now in this plot, each dot is going to be an individual participant. If they're towards the high end of the x-axis, that means their moral judgments were much harsher for intentional than accidental harm. So they were more forgiving of accidental harms. And then the y-axis is how distinct the pattern was in the right TPJ for intentional versus accidental harms. Those 2 things are very correlated across people. So people with more different patterns of activation made more different moral judgments. Okay, so that is an example of multivariate pattern analysis. It's an example of why it's so exciting because it gives us sensitivity to relevant dimensions for high-level social inferences. The example I showed you uses one approach to pattern analysis, sometimes called Haxby-style correlations. But the most general idea here, and this is maybe the punchline for anyone interested in fMRI, is a new way of thinking about fMRI data. So instead of thinking about, on each trial or each condition, how much response there was, multivariate analyses think of each trial as a point in the response to each trial as a point in the region's representational space, which is represented here as the space of voxels. So the idea is instead of thinking of the average response to this region averaging across voxels, how much this region responded, now what we think of is each voxel within this region is an axis, and that lets us treat the response to a story as a position in the space defined by the voxels. So if it was a big response on voxel 1 and 3, but a low response on voxel 2, that's a position in the voxel space. Once you think about fMRI data that way, right, then you can start to think about the difference between conditions as differences of the position in this representational space. And this way of thinking about fMRI data just opens up a huge world of possibilities in terms of what analyses we can do and what we can learn from them. So not just really simple cases like I'm showing you here where you're trying to decode a single predetermined binary dimension, but a much broader range of questions about the relationship between neural responses the continuous relationship, a data-driven analysis of that relationship across many abstract dimensions of the stimuli. Okay, so that is multivoxel pattern analysis. It is so powerful that it is definitely taking over the world in fMRI, and it is teaching us many things about theory of mind and about the brain. And then now the last question is, will it teach us anything about autism? So we have one small reason to hope that in addition to teaching us about how people think about the mind, it will also show us something about the mechanism of social deficits in autism. And again, that comes from the example of accidental harm. So we had found in a prior paper now a number of years ago that high-functioning adults with autism matched for IQ and education to a group of typical control participants gave atypical moral judgments, especially of accidents. So saying that Grace deserves more blame when the girl dies, even though Grace thought the powder was sugar. And what we found in this fMRI study is that the pattern difference that allows us to see the difference between intentional and accidental harm and predict moral judgments of that difference in typically developing adults was not present in this autistic cohort. And so the idea is that maybe even though the amount, whatever the amount of activation in a brain region is a measure of this kind of gross kind of processing that's going on, maybe the pattern information will be more sensitive to behaviorally relevant distinctions that are necessary for high-level inference. Okay, so the take-home messages from today. The first 10 years of fMRI of theory of mind using univariate analyses, fMRI was a very powerful tool to identify brain regions that play some role in theory of mind, that is to set up hypotheses about where to look if you want to study the neural mechanism of theory of mind, but was not, I think, very effective at revealing the mechanism of social processing difficulties in autism. The new tool that is taking over neuroimaging data analysis, multivariate analyses, is already providing rich new insight into typical neural representations underlying theory of mind, and may also be more sensitive to behaviorally relevant differences, including between typically developing adults and adults with high functioning autism. And that's it for today. Thank you very much.
Thank you, Rebecca. So we're about to start our Q&A now, and our first question comes from Lily Chaw, who asks, are the brain regions that are typically associated in theory of mind, for example, the right temporal parietal junction and the medial prefrontal cortex, associated with any other functions aside from theory of mind?
That is a very difficult question to answer. Because it depends on what you mean by a brain region. So the right temporoparietal junction and medial prefrontal cortex, those descriptions describe very large regions of cortex, right? There's a huge territory in the medial surface of the prefrontal cortex. And within that huge cortical territory are many, many, many brain regions that do many, many, many tasks. So for example, regions in medial prefrontal cortex are recruited for self-related cognition, for value-based judgment. There are regions that increase the response in proportion to value, and many other tasks. And the same for regions in right inferior parietal lobe and posterior superior temporal sulcus, each of which have sometimes been called TPJ. So the question is there a distinct region, the way we think of like V1, for example, a distinct neural population that might be defined by Sager architecture or by connectivity or by some other anatomical measure that has a distinctive anatomical instantiation and also a distinctive functional role? And the evidence is intriguing but not complete. Suggesting that, for example, the part of the right TPJ that plays a specific role in theory of mind seems to be a specific little patch of cortex that is distinct from other neighboring regions that do other things, like the region of posterior superior temporal focus involved in perceiving goal-directed action, and the region of right inferior parietal cortex involved in some aspect of attention and reorienting. But that has been very controversial over the years in the literature.
Okay, thank you. And our next question comes from Bill Martin, who asks, is the multivariate analysis on accidental harm that you were showing earlier based on the same data set in which you conducted the univariate analyses?
So the data that defines the features for the multivariate analysis, so when we say, where are we going to look in this person's brain to find their right TPJ, we use a simple belief versus photograph task, like the very first thing I showed you, in each individual participant, and we use that for feature selection. So we define a region of interest based on what we call a false belief localizer, and then in a completely separate task, extract the response to intentional and accidental harm. But we, of course, can look at the univariate analysis as well as the multivariate analysis in response to the same data. And I showed you an example of that. So if I go back to, where was I? So in this case, the univariate response to intentional and accidental harm that I showed you in this example is data from which we can decode in the multivariate signal, right? So here, there we go. So there I'm showing you the univariate response that is a big average response to stories about intentional harm and to stories about accidental harm, but not a big difference in the magnitude in the right TPJ. And yes, that is data in which the multivariate signal, so the pattern does clearly distinguish intentional and accidental harm, even though the univariate response does not.
Okay, and we have time for one last question, and this comes from Veronica Yuck, who asks, what do we know about the distinct roles of the different theory of mind regions? So right temporal parietal junction, medial prefrontal cortex, and precuneus and others in theory of mind processing?
Yeah, that's a great question. So many prior studies have tried to figure out what is the difference, in particular between the right TPJ and the medial prefrontal cortex, because those two brain regions are the most robustly and systematically associated with something like social information processing or theory of mind across a really wide range of studies and tasks and stimuli. And sometimes they seem to go together and sometimes they seem not to, and so there's been a very long, like a 15-year history of people trying to articulate what is the difference between the TPJ's role and the medial prefrontal cortex's role. And two kind of principal ideas have come from that literature. One is that the medial prefrontal cortex seems to think about other people in some way with reference to oneself. So in the medial prefrontal cortex, it matters whether other people are similar to oneself, whether they're familiar, whether they are close, whether they are liked. Whereas in the right TPJ, that kind of difference tends not to matter. The right TPJ cares whether people are coherent individuals, whether their beliefs are what you'd expect given the other things you know about them, but not whether they are familiar or similar or close to you. So that's one dimension, is whether other people are encoded relative to the self or not. And then another principal dimension, which might be the same but might be a different thing, is that the TPJ seems to be concerned with what I call epistemic features, so how you got your beliefs and knowledge, and whether your beliefs and knowledge are true and justified and reasonable, whereas the medial prefrontal cortex tends to care more about whether you feel good, whether you got what you wanted, so what I've called motivational features. So that whether the characters, what their motivations are and what their preferences are and whether those preferences and motivations are being fulfilled or not. So those are two kind of core principles that predict which brain region will be more involved. Just as one kind of neat example of this difference, there's actually been a number of studies that study the difference between intentional and accidental harm, but they manipulate what makes it an accident in two different ways. So in my lab, when somebody causes harm accidentally, it's because of a false belief, right? So Grace didn't want to cause harm, but more importantly, she didn't know that the powder was sugar. And it's her knowledge state, not knowing that the powder was sugar, that led her to cause harm. Whereas that when we do that, the information about accidental versus intentional harm is in the right TPJ. But other groups, including Jean Decidi's group, the way that they manipulate accidental versus intentional harm is that the action itself was caused without the character wanting the action. So for example, the person tripped and fell, or fell backwards, right? So their action was undesired. And when you manipulate accidental versus intentional harm that way, the information is in medial prefrontal cortex. And so my suggestion has been you can have an accident from not knowing, and then the right TPJ cares, or an accident for not wanting, or not controlling your actions by your desires, and then the information is in the medial prefrontal cortex.
Okay, I think that's actually all the questions that we have time for at the moment. And so just to let everybody know, a complete replay of this presentation will be available on the Spectrum news site in a couple of days. The webinar is part of an ongoing series, so please check our site for upcoming talks, which will be announced soon, and for video archives of previous webinars given by other speakers. If you didn't get to ask your question today, you can ask it in the comments section, which will be live on the page as soon as the webinar goes up on our site. Thanks again to Dr. Saxe, and thank you all for tuning in.